Okay, welcome everybody. So this is a special event. It is combination of the workshop, conference, physics and biology of proteins, and also of the local colloquium, which is initiative of the International Institute of Physics and of Physics Department at the UFRN. And today it is my pleasure to introduce Professor José Onuchic. José he started his career in the University of São Paulo in São Carlos, where he did his master's. Then he went for PhD, he went to California Institute of Technology. He was a postdoc at the University of California in Santa Barbara. And after that, he returned to Brazil for a brief period. He was a faculty at USP in São Paulo. And in 1990, he went back to the US. He got a position at the University of San Diego in California, where he stayed until 2011. During his stay there, well, José made himself as a very known scientist. So he worked in particular on the problem of protein folding, and he and his collaborators are responsible for introduction of the idea of funnels, folding funnels. So something that he will mention today. So José, currently he is a professor at Rice University in Texas. He is a member of many numerous advisory boards of different organizations, editorial boards. He is also a member of American Physical Society. He is a member of National Academies of Science in Brazil and in the United States. And his main interest beyond or generalizing the idea of protein folding is molecular biophysics and chemistry. Thank you, José.
Thank you, Dimitri. Thank you for the invitation. Thank you, the organizers, for getting me here. So the talk will be about two halves. The first half is going to be really, really general. So the people who do protein folding, I apologize. You heard that before, but it's part of a colloquium. So we go over the preaching about landscape. Okay, and the second part we tell a little bit about something new, and I think everybody should be able to follow, but it's going to be more interesting for the people that haven't been working on the fields a little bit more recent. So first of all, I want to invite all of you to come to Houston. We are in a very small university. Rice is a very small university. We have about a total of about 6,000 students, like 3,800 undergrads and about 2,500 grad students. So it's a very small school, but it's nice. And this is one of the new buildings. We are on the 10th floor of these buildings where you have our Center for Theoretical Biological Physics. That's a Physics Frontier Center. The National Science Foundation has 10 physics centers. We are one of the 10 centers, the one that's dedicated to the interface of biology and physics. So everybody is invited. It's a great place. Considering everybody that travels from the US to here, as you have noticed, I strongly recommend you to go through Houston instead of New York, Chicago, or Washington. So as long as you come through Houston, feel free to stop on your trip and come and see us. It's one of the most open airports of the year, so your trip is much safer if you choose any connection through Houston. So always remember that, okay? But although we are a small university, across the street from this building that's called the Bioscience Research Collaborative is one of the buildings created at Rice to make the interaction between us and the medical center. We have the largest medical center in the world. Here's where we are, and behind here is the Texas Medical Center that, just for comparison, has more high-rises than downtown Philadelphia. Okay, basically, if you go around this place, you have 50,000 people that are either an MD or a PhD working there. Just the Cancer Center, MD Anderson, has 2,000 PIs. That means 2,000 laboratories, and each one of them comes with a total of 220,000 employees. So we have lots of collaboration. If you want to interact with biology, that's paradise. It's basically, you have everybody work on any problem you want to there across the street. So if you are interested in these areas, please come and visit us. Okay, so now let's talk about protein folding. Let's go to the old story. I think we have people here. I think basically Angel Garcia is here, Hussein Chen is here. These are the people late 80s, early 90s, on the times that basically there were 5 or 6 theoreticians working on the field. And basically was an interesting field. It's nice to see the growth of it, okay? So protein fold became a very interesting problem. And it's very interesting to look at it because everybody say, who is going to solve the protein folding problem? And it's an interesting exercise if you do. If you go to the web and you check who solved the protein folding problem, there are people who have solved it, like Harold Shiraga, for every year for the last 50 years. There are people that have solved every 5 years. There are people who have solved every 3 years. So the question is, why is not everybody happy? The reason for that is, in order for people to agree on an answer, first they have to agree on the question. So we have to define what we call to solve the protein folding problem. If we have different definitions of the questions, then you're not going to agree on the answer. So for example, if you talk to a wet biologist, typical guy on a biology lab, they're going to say the proteins fold, therefore there's not a problem. Okay, and if you talk to someone from structural biology or someone doing biochemistry, they're going to say the protein folding problem is a problem where given the one-dimensional sequence of a protein, right? The protein is a necklace of beads, right? Basically just open necklace of beads. And you have beads of 20 colors, and you have this one-dimensional chain. What's the three-dimensional structure associated with it? But if I tell someone that that sequence has a well-defined structure, I'm already telling you a lot of information, right? Basically, how do you know? So when people came from theory particularly, and people from physics, theoretical chemistry, moved to the problem, the question we got interested is, what is, given a sequence of amino acids, if I give you a sequence of amino acids, can you tell me if that sequence folds? So what differentiates something that behaves like a protein that's able to self-assemble in a unique structure, and what's the difference between that and a random heteropolymer that's going to be there? So as we do everything in physics, we try to simplify to start. So let's think about our necklace of beads. There are 2 amino acids, but there are too many for us to do analysis here. So let's simplify by saying the basic partition of them, hydrophobic and polar. Okay? Hussein Chen made a career out of these two, use just these two. Okay? So you can actually think about a protein like that. Okay? So let's think about 2 colors. So I have a necklace of beads of the 2 colors here. And I put half of each color. And I have a very simple potential here. Since they are hydrophobic or polar, oil and water, I'm going to say beads of the same color attract each other, and beads of the different color repel each other, just to make a very simple potential. So what happens? When this protein tries to fold, what tries to do? Basically, everybody here comes from physics, you think about it. The beads of the same color try to get together. The other ones try to be apart, so you try to phase separate. You try to get a phase separation just like when you mix oil and water. However, you cannot fully phase separate. You cannot fully phase separate because different than oil and water, when you mix them, these 2 things are connected by a chain. So what happens is, although this object globally is going to try to phase separate, you're going to end up with a few contacts of the wrong color next to each other. Right? So you're going to get with a few unhappy beads next to each other. So that's a word the physicists call frustration. That's where the word frustration comes here. That means when you're trying to minimize the global energy, you end up with some global energies that are not satisfied. You cannot satisfy globally and locally the energy. That's the idea of frustration. And if you get a random sequence like that, and you perform this exercise many, many times, you're going to have lots of low energy states that are structurally different. And they are structurally different because each exercise, where they form the wrong contacts, are different places. And you have a landscape like that, what we call a random landscape. Okay? And this becomes a nightmare to find the lowest energy state of this landscape. But not only this is a nightmare to find the lowest energy state, even if you find it, it's irrelevant. Why? Because if I change a little bit of the conditions, change the temperature a little bit, or change the pH, or do one mutation, maybe this state come up and this one come down, so it's very non-robust. So proteins not only have to fold reasonably fast, they have to fold reliably to a state. So it has to be a robust state. So the way we do that is based what you need to achieve is you have to get one of these states and you have to make it more stable than everything else. Okay, so you want to drive that state towards a minimum of energy, okay? But you want to make sure at the same time you drive that state to lower energy, you want the states you're competing against to get smaller. So what you have is a system where you try to maximize this stability gap in units of how deep are the traps, which are the states you're competing against. Because a structural problem is a competition. Which state you go to versus the state you're competing against. Now, this ratio makes life of everybody extremely miserable. Right? People doing structural prediction, they're very good about looking stuff that gives good stability, but they don't know that different proteins may have different roughness. So the fact is, you can formalize as a ratio, but makes life very... And basically, the literature, the physics literature has lots and lots of papers by basically working on these things, with models, with correlation, without correlation, with many different things. So there's the entire literature. But the bottom line is, that's what Peter Wallace called principle of minimal frustration. And when we came to the game, we say, how do you achieve that? So the way you may think about how to achieve that is the following situation. Let's imagine I have my protein here, my polymer, okay, in front of me. And let's imagine I could enumerate every configuration of that polymer. It's a complicated thing, right? Basically, every possible shape of that polymer, you can just put on the shape that you want, okay? It's a tough numerical problem to do. Nobody can actually do. We did that on some lattice models on the early days. That's how a lot of lattice models came to the game, because there are things we could enumerate every possible configuration. And now, I'm going to get all these configurations, and I'm going to order them by saying how similar they are to the native protein. Now, we have lots of mathematicians here. We have been discussing knots and connections, but I can tell you one of the main things that you have in the problem until today is which are good measures of similarity, what mean this measure of similarities, how they associate to dynamics, to functions, so that's a big game. But let's assume that basically our favorite coordinate is Q. Let me tell you two typical coordinates. If you talk to a crystallographer, their typical coordinates is RMSD. It's root mean squared. They just put the structure to structure the top of each other, try to align them the better way possible, and you figure out the square root of the square of the distances of every atom on that protein. That's a good measure. Works in some ways. But in some cases, you can go wrong, because if you look at me like that, and you look at me like that, we have very different RMSDs, and we're still the same person. Right? So sometimes it's good, but sometimes it can really fool you by putting that. The one we have Q is one that's more energetic related, and that measures if I look up a structure of a protein, and I look all the tertiary contacts. The tertiary contacts in a protein, for people who don't know, are the contacts where two amino acids that are very far from each other on the sequence get physically in touch. It's a non-covalent contact. If I make a list of all the contacts define that structure, the Q tells me what percentage of that contacts on every structure I have here are similar to my low energy state. We believe that's a better coordinate for fold. It's not perfect. There's no perfect coordinate, but it's one we have been using. So, but let's forget about. So there is a coordinate I measure similarity. And now I put every state, and I look how similar they are to my native state. So clear you have your native state here. You have a few that are very similar. And then you have more and more and more and more states as you go up that are very, very different. How would I get a sequence that's able to do that? So the ideal funnel tells the following thing. Let's now make a geometrical assumption. A geometrical assumption is, it's a very important assumption because I'm going to break it very fast. So let me make it so I can break it. I'm going to assume that every state that's similar can interconvert very easily. That's not true, because in proteins you cannot cross chains over chain. But let's assume that you are in a geometrical thing, that this is not a problem, that states are similar. If I have this situation where states are very similar, very close, then I develop the following model. I develop a model where I try to make all my native contacts attractive. So every contact that's on native state is attractive, and every contact that's non-native repulsive. What happens in that case? What happens in that case, you see that now, you start up here, you make a contact, makes no difference which contact you make. You start to come down, then you make another one, make another one, make another one. You're going to have any factorial different pathways bring you down. That's the ideal funnel. If you had a protein like that, where you have no geometrical problem, and all the contacts for the initial state are attractive or repulsive, they come down. That's basic what is defined as an ideal funnel. That's not reality because, first of all, you're unable to make all the situation where all native contacts are attractive or are not, and the geometrical problem doesn't allow you to have all these things connect. Actually, geometrical problem is a much bigger problem for the protein than it is the energetic part. Like Joanna told you, proteins really avoid to make more than thread in one time. They try to get out of, they don't overdo their geometrical trouble unless they really need to. The structure you have on proteins tries to stay on geometries that you can reasonably do on this approximation they have. If that's the case, what happens? You have any factorial states. So if I have any factorial states, I could think about it. That's the typical figure of a funnel that has been around for a long time. And this funnel is a cartoon. It's the same figure I had before. Instead of drawing a bunch of states as a bunch of balls, I put as a width of the funnel. And the width of the funnel is the log of the number. Instead of just being the number, it's the log of the number because it's going to be an entropy term that we call configuration entropy. Okay? And here's energy. This energy, by the way, is effective energy because the solvation effects are included on it. So it's not a real energy, just for. And then I believe I have my n-factorial pathways. Typical funnel for a protein, you get about stability of about 1 kT per amino acid. So a typical protein is 100 kT. And the typical traps you have are 2 or 3 kT. They're experimental numbers. That's how proteins live. Now, if I do E minus Ts, now I can have several. If I have a system in very high temperature, I'm going to be populating the high energy states, right? Because I have many more of them. If I lower the temperature, I'm going to populate the low energy states. The folding temperature is the temperature where you're going to stay 50% of here and 50% on the unfolded basin. We try to associate that to all non-transition, a first-order transition, but it's a finite system. So we say it's alike. It's not an infinite system, okay? And actually, people love to do situations on proteins where they send n to infinity. It's always a bad idea. Proteins, typical proteins, have a very well-defined surface to volume ratio, and a lot of the properties of them depending on that. If you have an infinite system, what you have is a bunch of beta sheets collapse, and that's why you get fiber and stuff like that. So the bulk limit, also we love to do that. We all have Royd problems. That is a very problematic problem here. The surface to ratio on proteins is a very important thing that if you think logarithmically change very little. Okay, now, if I plot this free energy now at this folding temperature, I have a minimum, that's my unfolded minimum, and a minimum, that's my folded minimum, and a barrier in between. And this barrier comes from the non-perfect cancellation between energy and entropy. Okay? Here I have entropy, here I have energy, here I have E minus Ts, right? My barrier comes. Now, there's a very interesting thing here. This barrier is very non-robust. Victor Munoz can tell you that. That's why a lot of people work very hard on removing that barrier to make very fast folding proteins. Okay, so it's not a very important property of these proteins. But the most interesting thing is that if you buy my discussion now, if people experimentally try to understand folding by looking what happens at the top of this barrier, what you call the transition state of this reaction. So if you believe my perfect funnel told you that I have any factorial pathways coming down. My barrier is halfway through it. So if I look at the ensemble of states that live on the top of this barrier, they all should be about 50% native. It can be 50 or 60, but let's assume 50% is halfway through. But if all the pathways are equivalent, then if I measure the probability of every contact to be formed there, statistically it will be formed 50% of the time. Because all these pathways were equivalent. That's the ideal funnel. That's not true. That's what we call an entire homogeneous transition state. Proteins do a very good job on that, but they don't do a perfect job on that. Okay? So that was the idea. And people actually did experiments on what we call 5 areas. I won't go on details here. And the other things to measure what's the probability, the nature of that transition state there. What people do, they mutate every residue on the protein, and you observe if they affect, they know they're going to affect the stability, but you also notice if they affect the barrier. So you know if they're already structured on the barrier or not by doing that. If they're not structured, they don't affect the barrier. They affect only the stability. So this game, you can measure what the probability to do. Now, when we got the problem, I told you before, we didn't believe that the geometry problem, as I told you, is that simple. But we believe the proteins were meaningful state, they had a funnel. When I tell you every contact on the structure has to be native, that's not true. If there are about 50% of them, that's more than enough. You have just to have a strong bias toward that state. However, to prove the point, we developed what we call structural-based models, also people call Go models, that are these artificial models of the proteins, where we make all the contacts native. So in the year 2000, we say, let's check this idea. How we do that? I make a model, I assume my protein is funnel-like, and then I compare to experiments. So these models were great. They're a family of models. I shouldn't have to talk then about it, but United Airlines didn't bring Paul to introduce the models for me, so I'm going to give you a very light introduction on these models, okay? But these are models where basically you have your protein. This is the C alpha version of it. That's the low resolution, but you have an all atom version. They're all possible versions of these things. You say that the angles and the bond lengths are pretty much conserved. That means they have very strong harmonic constraints. And then the terms that really can move are the contacts between two residues, what I call here the RIJs, and these dihedral angles that you have 4 amino acids moving together. And actually, a good protein, I can tell you that we know from experiment, they sort of tend to divide the energy on dihedral angles and contacts. That means proteins have secondary structure where they form helices or strands, or they form tertiary contacts. Proteins don't, if a protein forms the secondary structure too early, they form it too early, then you have these big monsters on the way. It's very hard to organize them. You become geometrically in trouble. But if you form them too late, becomes a surge nightmare. So you actually want to form both together. Energy have this idea where these energies are equivalent. They all really play together. They cooperate with each other. So our models work on that. Paul will give you a lecture on that, so I'm just going very fast about the next slide. There are enormous quantities of models that can be used at all shapes of resolution. Anybody is interested, you're going to have 2 or 3 lectures on it, where it's going to tell you how to use these models, how we have played with protein folding. But there's a great database that we maintain together, has been built for many years, and we use to create these models. Now, what we did? What we did is we got some proteins, and we said, let's check for our sample is correct. So there are a few proteins that have been studied to death. You heard about CI2, trypsin inhibitor 2 by Victor today. This protein has been mutated as many ways you can. There's data by many, many groups, particularly Alan First, but there are other people that measure these things. They have been looking at the transition state of that. So we built a structural-based model and say, does that agree with what people observe experimentally? So let me tell you what you observe when you do the simulation. When you do the simulation, the protein, here are 2 proteins. I'm going to tell you study. This is CI2. This is SH3. Okay? So let me tell you a little bit about these proteins. This protein, let me tell you what this figure is showing you first. Here's the protein. This figure is what we call a contact map. A contact map tells you on every place where I have a point here, forget the colors. Don't use we come to the colors in a second. Tells you it's a contact on the native structure, okay? So every point here is a contact on the native structure. Means the 2 amino acids come together on the native structure. The color here tells you, are these contacts formed in the transition state or not? So I go to the transition state and I look. So dark contacts are likely to be formed, and light contacts are not formed in the transition state. Okay? If I have a good biochemist here, someone like Victor, he knows CI2, every amino acid. But for limit people like physicists, blues here means where the beta strands are, and reds where the alpha helix are, so at least you can have an idea what's making contact with sweet. So it's just to help your eye. So you look at this figure, and I tell you, this protein is a great funneling protein. And you say, wait a second, you just told me 2 minutes ago that if the protein has a good funnel, every contact should exist at the transition state with about 50% probability. What meant this entire thing should be about light red, maybe 50% is too much, and you have all possible different colors here. Then I say, give him a break. Give him one more chance for me. One more piece of physics. We're going to say that contacts that are closer in chain, they are easier to be formed than contacts far on the chain. That's loop entropy, okay? Just floor entropy, loop entropy is easier. So it tells you that actually not only this protein is full like a funnel, but the contacts that are closer tend to form first, and the other ones come after, the others come after. If you believe on that, you see that's exactly what CI2 does. Contacts closer diagonal, they're all colored the same way. As you go away from diagonal, they get lighter and lighter. But if you look around the diagonal, it makes no difference from here, here, or there. They can start to form by different ways. All these parts are involved. You don't have a part of the protein that forms first. So CI2 is really funnel-like. This is a protein, like you heard from Victor, you can beat as much as you want this protein, and it folds. When we got this result, we are extremely happy. We said we are fantastic theoreticians, and we're glad that we published that a lot before many people did, because we agreed with the experiments. The experiments were out there, and they said the transition state of this protein has an alpha helix that you see here, and they have a mini core that this part coming together, and we got exactly here. So great, we have an enormous amount of power, we are great theoreticians, and that's absolutely incorrect. All the credit here comes to the protein, not to the theoreticians. Okay, this protein has such a bias towards the native state. Right, they're such good folders that as long as you have a model that's reasonably sane, you are going to fold that protein. Okay, that's why people can fold proteins with big computers. It's not because they are smart, it's not because the force fields are good, it's because the proteins are great folders. Otherwise, you wouldn't be able to fold, even with all the computers in the world, these proteins that you have now. That's why you have these pathetic force fields, as we all know, and we still can fold these proteins. Okay, so that's a good thing to remember. We were happy we were first, but then every theoretician that came into this problem afterwards, they got the right answer. Because there's very little you can do. Because remember, that's the basic idea that comes on this landscape of the proteins. If that gap is large compared to everything else you're competing, you have a lot of room to get in trouble before you go to a different answer. It's a very strong memory towards that. Now, just for curiosity, this is a protein that's not the case. This is a protein called SH3. And this protein, as you notice, different than CI2, they have contacts that have to be formed early. So the contacts very far in the diagonal, they have to be formed early. And that's a geometrical requirement. So that's what we call a non-funnel-like characteristic. But there's nothing the protein can do. They're geometrically required to go there first. Right? It's like a knot protein. You have to thread. There's nothing you can do before you fold. So that's a requirement of the geometry. There's nothing I can do on the potential of my energy to do that. And that agrees with experiments again, what made us very happy. And we know that these facts, that this context to be formed early, has nothing to do with the energetics, because our model is a structural-based model where we remove all the energetic frustration. My energetics is good, so that's a geometrical situation. So when you look at that, we are happy, because if you observe this protein, and that time, we have Angel here doing big simulations, Joan is doing reasonable size simulations. That time, at that time, we did the first biggest simulation you could do for SH3. Scripps Research Institute got a cray, was an enormous cray. Charlie Brooks controlled that cray. John Shea was a postdoc on the group at that time. And we ran this protein with one of the most, it was a very clever simulation that today nobody's going to accept. We pretty much unfold that protein, create a bunch of states, and simulate around each point on that unfold trajectory, try to build a pathway. But the beautiful part is, and here, since we are doing a plot in 2 dimensions, we plot with a lot of data, we have two dimensions. My Q, like I always described to you, right? The same native context, and the range of gyration that tell you how collapses this protein. So we could separate the data a little bit, right? So it's just an extra coordinate. And when you look this thing, you observe that the following situation. You get a transition state that requires to form this context here early. And actually, when we saw that, we say, is this in agreement? How do we put all these things together? Then what happened is Margaret Chung in the group, at that time, Angel was collaborating with us, putting all atoms together. So basically, notice that this protein, when it folds, it forms these beta strands very early. Like I told you, the short contacts are formed early. But after that, you have to bring what you call this distal hairpin that's formed together to the diverging turn before you form the core. If you form the core too early, you are in trouble. There's some posters here of people going even on details of these possible pathways, but this was the story these days. So you have a geometric constraint that tells you, unless you form that state, you don't have that situation. So we're very happy, because you could show geometrically with our model, we are in exactly agreement with the experiments. You start unfolded, you form those strands, you have to bring that diverging turn and distal loop together. And then you have almost a fold protein. And the final state is that you have a protein with a hydrate core, and you have to expel the water out of the core. This is not in our model, but we actually work on that problem. That problem is, say, can we incorporate these two parts? So that's a very important thing that we understand on protein these days. Also, people like talk about hydrophobic collapse. That's correct. Proteins don't sort of form a dry core and get structure. They form a wet core, and then they expel the water. And these states, what we call dry and wet states, are there, and they're actually very important for the function. Otherwise, they won't be able to have ligands coming in and out. So there is water that goes to the hydrophobic core, and people in physics can understand, because you open the hydrophobic core a little bit. It may not like water, but likes vacuum much less, right? So the entire world is all relative, right? So there's one thing you don't have is empty space. That's very costful. So continue on that. With Angel and Margaret, we did a model. It's one of these nice models we have done these days. People have been talking about it. Angel was the first one to do the model. We just got it and put it into proteins. But the idea is, if you have two balls inside of water, hydrophobic cores, you observe what's the potential between them. Yes, they tend to have a minimum here, but they have a second minimum when you start to bring them apart, and you create a vacuum between, then they want to put a water in between. And the beautiful part of this thing here is, if you apply pressure into the system, when you get to about 1,000 atmospheres, for reasons I won't discuss today, this minimum becomes lower, and you get what you call pressure denaturation. That's why proteins under very high pressure, you actually can denature the protein by putting water there, and that's a number you can compute. I can tell you temperature effects are much harder to compute, but pressure one is able to compute because it's more local than temperature in a certain way. So what you observe here is now we create a model that basically not only you fold, but you have this dissolution. And people can ask, what's more important? What takes longer? It makes no difference. You need both. You have to be a fast folder, and you have this dissolution. And actually, it's very cool because in 2003, with Angel also, we're actually able to do a distant simulation of a folding of a protein. Now the world's done, but that's the first one where you could say an honest simulation. That was something done with replica exchange. Those techniques are starting to come to the game. So it was a great time to do these things because you didn't have to compete with anybody because you're just doing it first. So now everybody's trying. So basically, we are able to observe. And the cool part here is, here is the RMSD, and here's the Q. And observe, this is what we call the wet minimum. And here's what we call the folded minimum. And you see both coming here. And you see around TF, they're actually very similar. If you just play with the temperature a little bit, you can switch between open and close this protein. You have a competition. It's a very interesting story. If you think about it, the water sometimes, when the water comes inside of the protein, even on a hydrophobic core, although energetically is unhappy, it has more translational entropy than the water in solution. So it's a very detailed balance when you compute these numbers. A very interesting physics to do on that. And when we work on the game, we could actually count how you come. The first transition, you lost about 15 waters. The second one, you lost another 4 waters before you actually fold. These numbers could actually be computed from scratch. So this is the history. Introduction to protein folding in a certain way. Just to finish this introduction, and I think that's about half of my time, then I go to the second part, is the fact that basically these ideas work for a large system. People say, oh, these only work for 2 systems. No, these structure-based models work for many, many larger systems. We actually have done larger systems for many things. The first ones we did are systems here, 3 proteins that people knew they had intermediates, and you just find them. The intermediates just depends on this relationship between entropy and you find them on the simulations. The more interesting one here is QI, because this intermediate, actually, if you look very carefully, is a trap. You find it, but actually you have to unfold before you fold. And that agrees with the experiment. And actually, it's kind of a cool story, because you see here, you don't have a trajectory, you go from fold to unfold. You actually have a state in the middle, right, between Q that you're moving in between. So QI is this protein with all this alpha helix around the core of beta strands. This misfolded intermediate means they form the helix too fast. And if they form the helix too fast, they cannot fold. You just become a trap with the core disordered. So what you have to do is a phenomenon I call backtracking. These 2 helices have to unfold it, and then the core has to repack before you can fold. So these things are able to explain many data, and these models have become very successful, and they're moving along these days, and there are several variations of it. You have here about structural-based models. So they're models that basically, you're not predicting the structure. But if you have the structure, you can talk about mechanisms. So what I told you here, I didn't tell you anything about structural prediction, but I told you 2 things. I told that proteins have a funnel-like landscape on minimal frustration to fold. And I told you that if you have the structure of the folded state, I can build the entire landscape, not only for folding, but we have learned these days you can do for function. Yeah, that's great, but there's a trouble. That's still another part of the story, a more recent part of the story. There's a trouble here. The problem is, great, but what happens if you don't have the structure? So in recent years, we decided to look at this problem. You can say, come on, get more crystallographers, there are more NMR people, get more structure, go there and do it. Yes, that's part of the game. But the other thing is, can I get information from other sources? So that basically what we have limited information. We don't have enough structures. So what we came is, can we get information from genomics and use that information to help us to get structural information? So I'm going to present a little bit of that just for fun. I'm not going to be able to go too far, but I want to tell you the fun part, as people have learned today or during this meeting, people are not here, I can show the fact, that if you look in evolution, there are certain motifs that appear all over evolution on different stages of evolution with the same folding shape and complete different sequences. So what happened is actually, by selection, as I told you, proteins can only select folding motifs that they're able to fold to. They can deal with some geometrical problem, but not too many problems. So it's not like you can make a protein with any shape. Because they have too many geometric constraints, they don't fold. So if I believe on that, can I use this genomic information, the fact that I know a lot of the sequence, if I know that some proteins are the same proteins for functional reasons, for other reasons, they have the same structure, but I don't have the structure information, but I have the sequence information. Can I use the evolutionary information to try to get structural information? So this is a game that started with 2 great postdocs in the group, Martin Waite and Farouk Morkus. Joanna joined the effort at some point. I'm going to show one slide from what she did on this thing, on trying to get this information into the game. So what was the idea behind it? Terry Hua at San Diego started with me, so the first paper was with him. Now we have lots of other things without him, but he's doing his own. So it was really a team effort of our center to get these ideas moving together. So the idea we get here is the following. The idea we have here is that we believe that instead of looking, if I have a bunch of sequences that follow the same structure, I'm not going to look into sequence conservation. I'm look at covariance. I'm look how two residues move together. How, for example, this SP move into NI. So I look at covariance. And why is that? Because my idea is that if you have a protein and you have a disrupt mutation, you're going to have to select a sequence that compensates that mutation in order to make the protein stable again. So if I'm going to identify that in evolution, the things can be replaced by different pairs, by doing covariance, I should be able to find the pairs that move together, and if they move together very frequently, that's a good identification they are probably a contact in 3 dimensions. So we went after that, and let me skip this slide. It's just some rationale why this is being done now and not late. But the idea is we create this idea called DCA, where you align the sequence and find these pairs. The only difference what we get here is what we call direct information. And direct information means there are contacts that interact directly. They interact directly, but not contacts interact by another amino acid. So in order to do that, we have to get the global probability distribution of the system, not just mutual information. Mutual information will give all correlation between 2 pairs. We try to separate the local from the other one. I will tell you how we do that. So what's the information you have from data? Information you have from data, you can tell you how much a particular amino acid is conserved in a position after you align all the sequences from a particular structure. Normally, you need about 1,000 sequences that are independent in order to get good data, but that's a number that Martin Waite is actually working very hard on making this more. So I figure out the residue is conserved, just to make sure the weight is it. And the other one is you just figure out what's the probability of a particular pair exists in a point, and you do that for every possible pair. Okay? Garrick's here. He knows Michael Fisher quite well. And Michael Fisher always keeps telling us that statistical physics is just counting. It can be complicated counting, but it's just counting. And that's what we are doing here. We are just doing a lot of counting. So you're getting all possible pairs. We do all that counting. That's all we get together, right? You do all this counting. And when you do all this counting, you're able to come with something that has these 2 things. So what are you going to do here? What are we going to do here? The information we have is what? Single residue or pair interaction. And now I'm going to try to get the best global distribution because I want to get direct contact that satisfies this interaction. What do we have? Pairwise interaction and single point. That's what? 2 spins interact with each other, a single field. So what I have, a Potts model. That's where we are back to our origins. We have been done that for a lifetime, okay? If you have done statistical physics, you have doing spin models. The difference here is a Potts model is just like an Ising model, but instead of having a spin of 2 states, here you have what? 21 states, because I have to have 20 types of amino acid plus a gap. Okay, but the same thing. Now, so if I do that, remember here, this is using maximum entropy. Maximum entropy tells you that's the best probability distribution you can get. Give that information. Now, the question is, the best sometimes not good enough. You're losing important information. If this thing was important, if higher order terms were important, I would be in trouble. Right? But here appears the pairwise information. Proteins are really pairwise, gives most information you need. And Martin Waite and Farouk figure out the question you do now, you have to do an inference problem and try to figure out all these parameters back. What's hell of a job, and couldn't us make large sequence because it was very compute intensively, until they decide to play with the Hamiltonian a little bit, and they show that you're happy to solve this problem in a mean field approximation, where each spin saw the average of the other ones. You could show that these parameters that are here, these EIJs, actually come from the inverse of the covariance matrix. So suddenly a problem that was a nightmare inference problem became a matrix inversion problem, if you're happy with perturbation theory. Our experience these days is that this is not perfect, but consider all the other problems you have, this is a very good approach. So with that, we have been able to go to many, many, many different problems. So just to make people a little bit happier, you can show that basically you have this. The Ij comes from the inverse of this matrix, and the CIBG is just the Fij's minus, okay, just to remember what this matrix is, just compensate for that. So this became a great method to find contacts of systems that you have without systems. Let me show the results a little bit, because they're kind of cool under this spirit. So here's just to finish, is the following situation. You have this, and you try. Now, this Dij tells you what's the probability of a contact, and this is just P log P, as you all know, just controlled by the density and the fact that you're just getting the direct contacts on it. So it's just standard. Di is a good measure if a contact's likely to happen or not. This became a great tool, and there's lots of people using here. Even people like Dave Baker, he's borrowing these things into their models now. People are talking about it. Everybody's borrowing. Why? Because you have a structural prediction model, you don't have to replace your structural prediction, you just have additional information. Right? Every additional information you can add help your case. Right? If I have a good prediction model, but I can constrain a few long-range contacts, I'm in good shape. Those are difficult ones to get. So this is becoming a great tool to go there. So, for people's curiosity, here is a structure of proteins, basically. This is a paper that Joanna was involved, or people involved. It's kind of cool to notice that first, the Di pairs do much better than just mutual information alone. We don't know yet why. I think I know, because this global information comes in a better way. They take the information as a whole. But not only they do better in a sense that basically here, what's telling you is how many of these predict pairs are correct. And you observe that the Di does high. Here is the typical mutual information. Here's some Bayesian approach. But this is much cheaper. But more than that, the contacts on the Di that are in red, the true contacts, they're much more distributed all over the protein than the mutual information. That's more important for structural prediction than anything else. Because you have a few distributed over the protein, you can predict structures a lot. So here we are using information from sequence to find contacts from there. The cool part is that you learn it. These sort of nice plots to tell you the power. And that's why. Why is this working? Because remember, proteins are not designed, they are selected. So this context, as the systems evolve in evolution, the sequences have been selected. They have the memory of what they did. So the landscape in evolution, as you have to be alive, you have to fold all the time, is going to project this landscape as you come around. Right? So they are related. If you have a funnel-like landscape, then the sequences that you have when you are evolving evolution, they project the spatial representation of the system, and the system works. So this is kind of nice, because here is your native structure, and here are the predictions from in the top of it, the context. You see the red are the good ones, and the greens are the wrong ones. But even the wrong ones are pretty close. Well, I mean, maybe it's an alignment problem or stuff like that. You have very few that are kind of lost on space here. So the system does quite well. But what's even more cool is the following situation. I'm showing an example that I like. Tells you that, as you can see, you have these structures predicted quite well. Now, okay, so this is, I'm going to go back one slide, I'm sorry. I just want to make sure I had here. This work that Joanna did in the past, and she showed that basically if you give me the secondary structure and plus DCA, I can, if I know the secondary structure, if you tell me where the secondary structure is, just the local structure, I can predict structures with 1.5 angles from resolution. So it tells you that's a real long-term thing can help you, and that's why lots of people are using these days. But the cool part is, structural prediction is nice, but there's something else you can learn from that. One thing we got very interested is this plot. This plot's a typical plot for a protein, but you can have other proteins with the same thing. What you observe here tells me what the frequency that contacts happen. So as expected, you have right contacts with high frequency. You have what we call water-separate contacts. You heard from the work with Margaret and how we did. Some of these contacts are water-separate. You heard from Garrick about it. They're well predicted. The method does quite well. Then you have a tail. No problem. You don't expect the world to be perfect. But what we got upset is, how about this bump here? I have no problem making mistakes because suddenly I have a bump. One thing we learned from the CA is that basically if you have a signal, something's probably happening. If you have a signal with a high probability, something's happening. So this signal was bugging us. Is that a mistake or are we missing something? So what we notice is this is actually a great thing. Because you work around this part, you start to observe, here's a structure. Here are two residues that should be in contact. Gives large DI, but they're not in contact. But if you work on that structure, it's a false positive monomer, they actually have a multimer. Okay? And these contacts are contacts that form three-dimensional structure. So that's the beauty of DCA. Now I can use what's so-called the DCA mistakes to predict multimers. And that's becoming a big game on protein-protein interaction these days. We start again, and the entire world is catching up on it. Right? It's a great tool to go after it, right? Because one thing we know is sometimes evolution doesn't give all the signals that you need. When they give you a signal that's very strong, very rarely they are there by mistake. Right? Some things conserve for a long time. It can be an accident. There's always accidents on it. So here we found all these different contacts, true contact, true contact, true contacts, we got it. Let's skip here. Now, here's something I like. The fun part is even for proteins, you have a crystal structure. Here's a functional situation. You may have a crystal structure of one of them. Here, for example, you have work that's done by Farouk and Bimanjana in the lab. You have a case where you have this leucine binding protein. You can have the crystal structure of the open structure and closed structure. This is one they actually had both, but in many cases you have only one. So here's just an example to show you. I'm going to show two examples, one that's trivial like this one and one that's interesting. So what we learn is the following thing. If you have a look, here's the open structure, here's the closed structure. Here's the difference in yellow between the open and closed structure. And if you have a look here, here's the difference between DCA. Here's a comparison between DCA contacts and the closed structures. DCA gets a lot of the closed contacts. So what we learn in a system like that is that now, if I run a simulation where I start just with the open structure, a structural-based model, plus DCA, I can find the closed structure. So it becomes a great tool, not only to find different structures, but also to find different functional states. Because they may exist, they appear on the data, right? So we're very happy. Here is just a comparison of put contacts at random to show if I have, you see here is the closed, here is the basically state, the open and the closed. So you will, it's actually the other way around, the way it's written wrong here, but you observe that you get the situation in there, right? So basically you see that here, you only get the open that tells you if you put this contact at run, this is just a control. Now, here's a more interesting one. Here is a DNA ribose binding protein. What's kind of very cool, because that's not a trivial one where it's just open and close. This is something that DCA predicts a twist intermediate, very far away from the open and closed state that you have. You find the open, the closed, and this twist state. And that's on the signal. And when you go to the data, there are a lot of papers, both from simulation and from structural information, predict that state, but they never saw it directly. So you can see the states more like that's kind of a cool state that still the information is in there. And they can find that based on the functional thing, the thing has to twist before the ribose comes in and twists back. Right? So this is a great tool. And on our Brazilian connections, we had a situation where Ricardo Santos from São Carlos was there. He worked with Adriano and Glaucius in São Carlos. He was there for his FAPESP during his PhD for 1 year. And he applied this method to find homodimers for all possible homodimers. And they create a great tool. They knew how to bring the dimers together. You normally put these potentials as in the physical size. They start with the potential very long range and very broad. And then with time, they reduce then the real size. So by playing with this scale using the DCA data, they could actually move these things into the right conformation without getting trapped on the way. So they had a great stuff on that. I won't have time to go on details. Here, they try to put this context, and here's what they have done. They even are able to find some domain swap structures coming out of these things. Right? So it's sort of a very cool thing that comes. Domain swaps are very nice because domain swaps have almost all the same context, but not all of them. So sometimes you can see if you have one or the other, and the method really helps you on that direction. Sometimes you have context that only satisfy when you swap. And that's a big thing. They are working on it right now. So we can do these things. And you see, we can do structures with very, very good level on finding structure there. OK. On my last 6 minutes, I'm reading my clock there, I'll tell you about a cool story just because it's cool. And then we finish. But I can do that in 6 minutes. So this is a story that tells you, I learned about these potentials to find structure. Can I now be so aggressive that I can say, you know, now not only I'm going to look at this potential for a structure, but if I know if a contact is native, and some pair of residues happen more often than the other pairs, can I develop a potential from there? If I believe the landscape of folding and sequence space the same, I can say pair of contacts more like is more attracts potential than one's less like. So I really squeezing the data. So we're putting this into the game. So we use that to do something what we call understanding bacterial two-component signaling, where you have a histidine kinase, that's pretty much something that phosphorylate, some response regulators, and they're all very similar. And how they identify the right partner. Can we figure this out just out of DCA, okay? So this is how most signaling happens on bacteria. There's about 100 to 1,000 of them in bacteria. The game is always the same. Something comes here, you have a signal get a phosphate to be there, you find your partner transferring there, okay? And then you go down into the world, and you signal something, and response regulator goes. So this is something you try to do, and now you can have this pair of proteins here, where you can do this situation. Can we play this game and try to understand this story? So here, we have to build sequences that involve both proteins. So we do a super sequence, the two proteins involved, and we look for correlation between them. We know how to line the proteins together, because in bacteria, most pairs on the same operon, so you know which ones are together, okay? But that's, you can get this situation. So now, what I told you before is I do the same thing, but in addition to just have a Hamiltonian, I actually have the type here, and I try to get an energy out of it. Right? So I do the same thing. It's a cool idea. You just say, and there's a lot of math here and calculation, but it's very simple. It just tells you if a particular pair comes open more frequently, they're probably more attractive than someone that doesn't go there. If I have enough pairs, I can do statistics and get a potential out of it, right? Just from likelihood of things coming together. The cool part of it is the following thing. This is something we are writing a paper now, but we have to be very careful. Is the following thing. If I get one family of proteins, and I compute delta G from these proteins versus delta G experiment, on the same family of proteins, I do extremely well. Okay, I do better than any method that's calculating delta G these days with force fields or with anything. Now, remember, I'm not calculating. Evolution is calculating. I'm just extracting the data. Right? I'm not doing any particular physics here. I'm just getting my potential that came from probability from the sequence information. And I can get, so this really gave us the confidence. These are different families of protein. If you try to put a universal potential, it's a problem because of the unfolded basin. But for one family, you can do it. So that gave the idea that's a great thing to study mutations, right? So I can do mutation to this system. And I'm showing a few slides just fast to finish because I know everybody's getting tired. So again, we do these things for these two systems. This just out of curiosity, we work on the system. We just chose the system. This is what you call sporulation versus competence. Bacillus subtilis it's a system where the bacteria decides it's going to sporulate when it's under stress, or if it's going to try to eat all the ones that already sporulate, the DNA, and try to change itself because it doesn't want to sporulate, but that's very costly for them. But the important thing is we are looking at this first step. Okay, this is the first sensor that met all the stress, goes to this kinase, and this goes to spot 0F. So when we look at this data, we can look two things. First of all, we look at the DI that I told you, that's the measure we do. That's what we measure from the situation. But we break this DI into two parts. One part, we believe that every response regulator does the same chemistry, so it should be the same. That's what we call our new model. We scramble all the sequences, and we take that part, and this other part left over is the cognition part. So when I break that, there are experiments where people actually mutate every residue on this spot 0F and observe how that phosphorylation change. And you see, forget this plot here, this is just the initial. Our method does quite well. You can predict when you get faster, when you get slower, you do quite well. So this is a sort of an interesting story, comes to the way of doing things. You can use this method now to do function. I have lots of other stories on function. I don't have time today. I just wanted to do a prelude of what's coming on other areas. So just give me 2 slides just to show a prelude. Let me go over this data fast, but I'm only able to show the other data. This story we don't have time to tell. It's a cool story, but I don't have time to tell. This is one of the things that's becoming a big game these days. It's going after chromatin. So these are the kind of models we are trying to do. One of the models is we are trying to create models where we identify how the chromatin has different types, and you try to get the structure of your entire genome. For the people that come from mathematics here, that's a fantastic problem. Remember that your genome is the largest molecule in the organism, is a molecule that's 2 meters long, okay, and has to fold into a nucleus that's 6 micrometers without making knots. Okay? So there, the mathematicians can have a field day. The field's a great word in mathematics anyway, so you can have a field day, right? So it's a great problem in terms of base. This is a problem where I think geometrical and topological problems are going to become much, much more important than they are on proteins. So I think that people can start to have the fun on it. We have a model where we start from chip-seq data, just to tell you. That's the kind of games we are playing these days. I'm sorry, wrong way. We start from chip-seq data. We do machine learning using our DCA approach the same way to figure out the types, right? These are histone markers that tells you from epigenetics which the parts, what's the different levels, which parts are functional, not functional, what genes are expressing. We predict the types, and we predict the structure. Okay, we have the first few papers on that, but that's a very hot field these days. So if you think about it, we are trying to use our copolymer ideas into this problem these days. And that's a long story. That's just to tell what's coming. We have the first few papers on that. Young people should have a look on this problem. It's a beautiful problem to work on. Try to understand how these things do, but we don't have time. I just want to thank the people who have been working for me in protein folding for all these years. As you can see, I wouldn't be able to do that if it was not for this fantastic group of postdocs. Joanne is here. Paul Whitford is arriving tomorrow, should be around. We have Vitor is here on this list somewhere. I can find here, there he is. It was a great issue with me. A lot of things got done. And I have to thank them for a long time. People always ask me, If you travel so much, how can you get work done? How do you get work done when you're traveling? I say the same work I do when I'm not traveling. The postdocs, the students do it. So in a sense, I want to thank for the funding agents. The NSF has supported us for a long time. And these days, we're very grateful for the state of Texas, the Cancer Prevention Research of Texas. So Texas created a program of $10 billion into cancer. And they believe a few theoreticians should get some of the leftover. And we get some money from that. And the Welsh Foundation that tends to give us money every year. So that's a way that shows we are there. And I thank you for your attention.